This is video number 1 in our series about myopia. In it, we're going to discuss what myopia is, how frequent it is around the world, and why we should care about it. Myopia, or nearsightedness, is well known as a cause of blurry distance vision. But it has a less well-known side, risks it poses to eye health. For example, it is associated with retinal detachment and a form of macular degeneration. For the last few decades, the frequency of myopia has been increasing around the world, in some areas reaching over 3/4 of the local population. And it is expected to continue its increase in the coming years. The increasing levels of myopia and attendant complications have motivated a search for why myopia happens and to find preventative treatments. It has become a major topic in public health. Our series on myopia contains 5 detailed subject videos. Number 1, what myopia is, how widespread it is, and associated factors that are thought to cause myopia. It will also serve as an overview of the series. Number 2, what risks myopia poses to eye health and vision. Number 3, research showing how the eye controls its own growth. Number 4, why do some eyes diverge from stability and grow into myopia? And 5, and last, development of treatment methods and their effectiveness. This is our plan for this video, Getting to Know Myopia. We dig deeper into what it is. We look to see who around the world has it. Then, how big a role do genes play versus the influence of the environment? What is myopia, more commonly known as nearsightedness? Briefly, in the sense that your eye works like a camera, if you had ideal vision, light rays from a distant object would come to a focus and make a sharp image on the retina in the back of the eye, what we call emmetropia. In people with myopia, usually it is because the eyeball is longer than average. One result of which is that light comes to a focus in front of the retina, making a blurred image at the retina. An example image comparing the emmetropic ideal versus myopic blur. The focus can be restored using a corrective lens, specifically a diverging lens to push the image back to the retina. Think glasses, contacts, or refractive surgery. The other result of the longer eyeball is that the layers are stretched thinner, causing problems in each of them. What then is the impact of myopia? The longer eye threatens vision loss from either refractive error or pathology. With a vision chart for perspective, on the right are criteria for vision impairment and blindness from the US and the WHO. They differ slightly. In 2013, the leading cause of blindness worldwide was cataract, and not far behind, uncorrected refractive error, which means myopes without distance glasses and presbyopes without reading glasses. Regarding moderate to severe vision impairment, uncorrected refractive error accounted for a little more than half. Such reduced vision impacts people's daily lives, like school performance and job productivity. Before we go further, a word about our approach to this subject. Even though myopia has been under study for several decades, it is still an actively developing field. Pinning down causation and deciding who, when, and how to treat are all works in progress. Because this is still a developing field, I've chosen a study-based approach, presenting landmark studies in major reviews, comparing and contrasting results, including disagreement between authorities. In the end, the purpose is to identify who will get a significant level of myopia and how to prevent that. We start with the basics, refraction and eye growth. As we said a minute ago, in the ideal eye, the image from a distant object comes to focus exactly on the retina. The cornea and lens in the front of the eye act together to focus the incoming light. In the business, we call the front of the eye the anterior segment. To form a sharp image, the focus distance must match the length of the eye. We call the back of the eye the posterior segment. At birth, the eye is usually relatively short, therefore farsighted. Ideally, the growth of the different parts of the eye are in sync, so we end up with an in-focus adult eye. In ideal vision, light rays make a sharp image on the retina called emmetropia. To the extent eye growth gets off track, the eye can become near or farsighted. The amount the eye is out of focus is called the refractive error. Usually, hyperopia resulting from a too short eye and myopia from a too long eye. The power and sign of the lens needed to bring the image into focus is called the refractive correction. Correction is measured in diopters shown on the number line. Hyperopia correction is in plus numbers, here to the left of 0. Myopia correction is in minus numbers, to the right of 0. Regarding myopia, some studies specify that myopia formally starts at a spherical equivalent of minus 0.5 diopters. Others go to minus 0.75 diopters. Most studies categorize minus 50 to minus 3 as mild myopia, minus 3 to minus 6 as moderate myopia, and greater than minus 6 as high myopia. WHO puts high myopia at minus 5 diopters. These divisions are based on both vision and complication rates. Note the color coding for low, medium, and high. We will continue that through the video. For those interested in details, the spherical equivalent refraction is usually used, here showing a sample calculation. This is what refraction looks like at age 3 months. Infants average about 2 diopters of hyperopia with a moderate range of powers. By 9 months, the gold line, the average is closer to 0 and the spread is narrower. This improvement is the result of the fact that eye growth is self-correcting by a remarkable process known as emmetropization. It is the subject of video number 3. How specifically does the eyeball change as it grows, and how does that lead to myopia? For that, we need measurements of the focusing system and the length of the eye. Respecting historical precedent, we start with an early study by Saw and co-workers who measured eye growth in children ages 7 to 9. They also looked at associations with environmental factors. These are the various parts of the eye that were measured. These are the changes with growth of specific parts of the eye and associated refractive error. The results are easier to see if we graph them. On the left, notice the eyes are becoming more myopic. The center graph shows anterior segment focusing power changed very little, while on the right, length increased. Conclusion number 1. It is increasing length of the eye that correlates with myopia development. In part 2 of their study, they looked at environmental factors to see if they were related to myopia and eyeball measurements. We will discuss that later in the associations section. Looking more closely at refraction in childhood, Ma showed that at the end of early development, emmetropia dominated. But then, around age 6, something changed and myopia started to increase. Jones gave us a more detailed view, looking at refraction behavior in groups, again showing a relatively narrow range through age 6. Then through childhood, emmetropes and hyperopes remained mostly stable, while myopes progressively diverged. Jones went an interesting step further, breaking changes down into the component eye parts. Note in this graph, myopia increases going downward. For emmetropes, the power of the cornea and lens gradually decrease, while axial length increases. In this case, synchronized so the refraction remains stable, near 0. For myopes, here the red lines, relative to emmetropes, corneal power remains higher, while lens power drops lower, which tend to offset each other. The big change is axial length, off the top of the chart, while myopia increases off the bottom of the chart, in agreement with other studies that axial length is the controlling factor in myopia development. The graphs showing myopes as a group suggest that all myopes diverge at the same time. But that is not the case. Myopia onset occurs at different ages. The earlier the starting age, the higher the resulting myopia level. And the divergence from emmetropia toward myopia actually begins several years before passing the myopia threshold. These highlights of myopia development are meant to interest you in video 4, further discussion of eye growth into myopia. We continue myopia basics with the structure of the eye and what can go wrong. In order to understand what can go wrong, you need to know about the structure of the eye. Here I have constructed a conceptual diagram representing the normal eye. It exaggerates the thickness of each of the layers, showing them as even from front to back. The innermost layer is the retina, a layer of nerve tissue that acts like film in a camera. Under that is the choroid, a layer of blood vessels that supplies the outer retina with oxygen and nutrients it needs to function. The outside is the sclera, a tough fibrous layer that gives the eyeball its structure. In myopia, as we will see in video 2, the lengthening of the eye results from thinning of the sclera. In turn, the thinned sclera is associated with thinning of the choroid and retina and their associated complications. First, the elongated eyeball results in refractive myopia. Second, the thinned, weakened sclera is also vulnerable to local bulging, called a staphyloma. Likewise, the choroid becomes thinner toward the back of the eye. This is associated with atrophy, part of myopic macular degeneration, here represented by a pale area in the central retina. The retina, on the other hand, has normal thickness in the back of the eye, but is thin at the equator. This peripheral thinning increases the chance of retinal tear and detachment. Lastly, changes in optic nerve structure increase the chance of glaucoma. That is, pressure-related damage to the optic nerve causing visual field loss. It is a key concept that the risk of complications increases with the degree of myopia. Myopia pathology is covered in detail in video 2. Now that we have an understanding of myopia and its complications, let us look around and see how frequent myopia is around the world. In 2015, Nature carried this news piece headlining an epidemic of myopia. I include it here because it is cited just about everywhere. Myopia is not evenly spaced around the world. These are the percentages of myopia for selected regions in the year 2000, ranging from a low of 5% to a high of 40%, with an average of 23%. Some local areas have higher percentages. This is where the regions are expected to be in the year 2050. The global average a little more than doubles, with regional highs over 60%. From the many studies on the epidemiology of myopia, we have selected 2 that report on frequency of myopia over time. Starting in the U.S., the Vitale study compares National Health Survey data from the early 1970s to the turn of the century. The prevalence of myopia at the beginning of the 1970s is about 25% across age groups. 30 years later, myopia has increased in all age groups. For example, an increase of over 18% in the 25 to 34 age group. The dashed line representing 50% of the population, and that was 20 years ago. Looking at it as a bar chart, again by age group, you can see the increased prevalence. But also note there is an increase in higher levels of myopia. In 2018, Morgan and others gave us a 2-part review aiming to put causative factors into perspective. In part 1, they assembled myopia data for these 4 Asian countries back to around 1940. These are the data points. We can connect the dots. Note the great increase in prevalence of myopia from 1940 to the present, reaching 80% or higher. Where did this large increase in myopia come from? That brings us to the big question. Is it genes, or environment, or a combination? Question 1, then, is to what extent do genes control myopia? Traditionally, questions of inheritance have started with twin studies. We take the genes in myopia twin study. You may recall that twins that come from a single egg are genetically identical, called monozygotic. Twins that come from separate eggs are genetically distinct, called dizygotic. Studying twins gives us insights into the effects of genes versus environment. These graphs show correlation of refractive error within the twin studies for the two twin types. Monozygotic twins have a very strong correlation in refractive error, whereas dizygotic twins are much less likely to have a close refraction. The same holds true for axial length, as well as anterior chamber depth, corneal curvature, and corneal astigmatism. By genetic modeling, they report the heritability of refractive error as 88% for men and 75% for women. This was the basis for the long-term dominant view that genetics controlled myopia development. Another way to get at the genetics is to search for individual gene defects. Keep in mind these are uncommon individual genetic defects that should be considered separate from the general population of myopia. The first myopia-associated gene to be discovered was MYP1, known as Bornholm eye disease. The resulting phenotype is specific for development of high myopia. It is interesting that it maps to a color cone defect. Up to 2010, linkage studies had identified 20 myopia-related genes. Since then, another technique, the genome-wide study, has yielded a great increase in the number of associated genes. In a genome-wide study, a few cells are harvested from each individual, which are used to sequence their genome searching for specific markers, single nucleotide polymorphisms, or SNPs. Those markers are then compared between healthy people and those with disease. If the markers are more frequent in the people with disease, it is said to be associated with that disease. It doesn't prove causation, but suggests there is a connection. Meta-analysis means combining results of multiple studies to try and get a broader, more reliable analysis. The genome-wide meta-analysis has become a popular tool for investigating a variety of traits. Among them, a genome-wide meta-analysis was organized to study myopia genes, run by the Consortium for Refractive Error and Myopia. Their first report in 2013 combined the results of 32 genome-wide studies, analyzing approximately 2.5 million markers from over 45,000 participants. In 2018, they issued a second report, increasing their data set and merging with 23andMe to exceed 160,000 in the new study. They did several things in their reporting. First, they developed a risk score using markers to predict persons at higher risk, at 40 times more likely to have myopia than those at lower risk. They determined which genes had the highest association with myopia and ranked the strength of association. These were the 19 genes with the highest priority scores. What do these genes do? The authors created a nice graphic showing where these specific genes are functioning. This is an updated diagram from a 2019 special IOVS issue on myopia. Digesting that into its basic message. The genes they identified as associated with myopia principally are involved in retinal cell physiology and light processing. The most significant gene was GNB3 in glutamate signaling. Also significant is the association with the dopamine pathway, which in animal models has shown a role in controlling eye growth in response to light. The author's general conclusions support the genetic involvement in light processing as a contributing factor to refractive error, a conclusion that agrees with current theories of emmetropization. However, before we leave the genetics section, we need to recognize a couple notes of caution. Genome-wide studies raised a lot of excitement, promising that by identifying an exact set of genes, or SNPs, they would be able to accurately predict particular traits. It turns out, identifying the effective genes is not so simple. For complex traits like height or myopia, identified SNPs generally account for only a small fraction of the phenotypic variation expected from twin studies. The 2018 Cream paper, even with a combined data set, only accounted for about 8% of the variation in refraction. That compares to twin studies which found around 80% heritability. That large gap in strength of association has become known as the missing heritability problem, raising a debate about the relative roles of genes, epigenetics, and environment. Commenting on heritability, one authority sums up the genetic contribution as more complex than expected. These are good references if you wish to pursue this. We now switch to the other side of the question. What about environment and myopia development? We know that genetic background accounts for basic susceptibility, but it doesn't explain the whole picture. Researchers believe the prevalence is increasing faster than can be accounted for by changes in gene frequency. There must be other contributing factors. In our modern world, children spend more time indoors with less exposure to sunlight and less exercise. There is increased near work, as in studying and computer work. And more recently, there is a question whether there is a contribution from the way standard glasses correction works. The next set of studies define parental and environmental associations. The SAW study we presented earlier found an association with parental history of myopia, reading more than 2 books per week, and other factors listed here. The p-values show association of parental myopia and reading with axial length and myopic refraction. Looking deeper, this study from Sydney, Australia was published by Rose and co-workers in 2008. They collected data on 2 age groups, one about 6 years old, the other about 12 years old, looking for connection between multiple risk factors and myopia development. Note there are more myopes in the 12-year-old age group because they are further along the growth curve. The graphs that follow are constructed from the Rose data tables. The first graph shows that having more parents with myopia is associated with more myopia in children, which fits our expectations. Next is outdoor activity. On this graph, the y-axis is refraction, with hyperopia increasing toward the top. Myopia is off the bottom. For both age groups, there is a significant trend, higher amount of outdoor time with a higher level of hyperopia, which is equal to saying higher outdoor activity results in less myopia. Regarding near work, the x-axis is hours of near work broken into 3 levels. In this study, hours of near work did not show a significant correlation with refraction. This 3D chart looks at both risk factors together, near work and outdoor activity versus refraction. If you view this by level of outdoor activity, within each level the amount of near work does not make a significant difference in the refraction. That is, in each horizontal row the boxes are about the same height, confirmed by p-values ranging from 0.7 to 0.9. On the other hand, within each level of near work, the outdoor activity makes a significant difference. That is, in the vertical rows there is a clear trend of increasing height with the p-values given. This is the published 3D chart from the Rose study. I'm including it because it is often referenced. Here the y-axis is odds ratio of developing myopia, and the direction of rows is switched. The orange line is an odds ratio of 1, meaning a 50-50 chance. It is clear that more outdoor time means a lower odds ratio of developing myopia. In addition to the main conclusions, the authors made a few additional comments. It was likely that outdoor time was the controlling factor, not sports participation. Indoor sports made no difference. Second, not only was there a lack of correlation with near activity, they also looked at mid-range activity, TV, video games, and computer time, finding no relation with the final refraction. Like the Saw and Rose studies, Jones was looking for associations of myopia with environmental factors. Part of their results presentation is an ROC curve. In looking for association between two factors, the nearer the plotted line is to the center diagonal line, the weaker the association, approaching random. Whereas the further the plotted line gets away from the center, the stronger the association. In this case, both parental myopia and hours spent in sports are significantly associated with myopia development. Reading hours show a possible association, but dropped out statistically when corrected for the first two, in contrast to Saw, who found a connection with number of books read. Similar to Rose, Jones found a lack of association with TV and computer time. Another part of their results presentation shows more myopic parents clearly means higher risk of myopia, and amount of outdoor time has a big effect on myopia within each group. A child with two myopic parents who spends little time outdoors has a 60% chance of becoming myopic. However, if that child with two myopic parents spends 14 hours a week outside, the risk drops significantly to the same level as a child who had only one myopic parent, and below the risk for the other ones who didn't exercise. Also note that children who have no myopic parents still develop myopia. Indeed, in the low exercise group, the risk is about 30%, the same as having one myopic parent. Author's conclusion, quote, "A lower level of sports and outdoor activity and having myopic parents were the best non-ocular predictors of future myopia in the premyotic third grader. Not so much with reading." In 2019, prominent researchers Zadnik and Muti published an editorial on their views of myopia causes, near work versus sunlight. Consider this as one side of an expert point-counterpoint argument. Zadnik and Moody identified outdoor time as important, specifically referencing the 2007 Jones graph. They did not find a convincing case for near work as a cause of myopia, but puzzling that light exposure does not slow myopia once it is present, referencing the following 2017 meta-analysis, which found that outdoor time is effective at reducing onset of myopia, but does not appear to slow development in children who are already myopic. Note, this is before the 2018 Wu result. For the counterpoint view on reading and near work, we turn to this 2018 review by Morgan. In a survey of the world's literature, they found that educational systems that require a large number of hours spent in studying and near work and allow for fewer hours of outside time show a strong relation to increased myopia development. A similar study on this theme was done in Israel. They studied 22,000 male recruits for military service. The school system in Israel is divided into levels which require different amounts of intensity and near work, with ultra-Orthodox schools requiring the heaviest reading/studying load from an early age. Relating prevalence and degree of myopia to school type, clearly the ultra-Orthodox schools had a greater degree of myopia overall, borne out by prevalence and odds ratios. Whether this is from more hours of reading or less time outdoors, or both, is not addressed in the study. There is one more factor among leading contenders for causes of myopia development. In the beginning, we discussed that eyeball growth is controlled by a process called emmetropization. It turns out growth is controlled by the clarity of the image not in the fovea, but in the mid-periphery. Specifically, it is peripheral hyperopic defocus that triggers increased eye growth and myopia. That is an important concept because standard glasses focus the image on the fovea, but may leave the peripheral retina hyperopic. This has become a major avenue of investigation for controlling myopia, motivating trials of multiple versions of glasses and contact lenses that aim to control the peripheral refraction. There is a lot more about this in the other videos. Now, we look at another approach to the genes versus environment question. There are large regional differences in myopia around the world. Why should that be? Higher gene frequencies in certain populations, or local environmental influences? How to answer that question? What if you look at children from the same country who presumably have the same genetic background, but who live in different environments? Specifically contrasting urban versus rural. In 2019, he and co-workers took a shot at that question by comparing published myopia rates among children in the urban areas of Hong Kong and Guangzhou, the semi-urban area of Shunyi near Beijing, and the rural area of Yangji. The orange lines show the rates of myopia in the two urban environments. Compare that to the lower rates of myopia in the semi-urban and rural environments in the same country. The authors concluded that there is an urban-rural difference, which is likely the result of differences in environmental factors, possibly including educational, socioeconomic, and/or nutritional effects. Another approach is to ask, what happens to children who relocate from China to Canada? Do they have the same rate of myopia as in China, or do they take on the lower rate of their new surroundings? For the China reference, we use the Guo study from Guangzhou. For the Canada reference, we use figures from New Brunswick at age 6 and Waterloo at ages 6 to 8 and 11 to 13. In 2007, a study was done by Cheng and co-workers in Ontario, Canada, looking at ethnic Chinese children whose families had moved to Canada. The rate of myopia was 22% at age 6, increasing to 71% at age 12, quite a bit higher than the Canadian population. If we overlay the results from Guangzhou on the Chinese-Canadian results, the myopia curves look similar. The Chinese-Canadian children retained the higher prevalence as in China. To try and figure out if this is genes or environment, Cheng did a questionnaire quantifying daily activities. The Asian children had significantly more hours of close work, not a significant difference in TV or video game time. They had significantly less outdoor hours, evidence suggestive for the influence of environmental factors. One more study on this question, this time focused on Singapore. They compared myopia prevalence among different ethnic groups who lived in Singapore versus the same ethnic groups outside of Singapore. There is a large difference in prevalence extending across ethnicities, with more myopia in the, quote, shared environment of Singapore, close quote, adding to the case for environmental influence. We conclude the section on causative factors with the 2015 CLEAR study. This large multi-center study looked at a wide range of factors to try and identify if they could predict development of myopia. These are the 13 different parameters they tested for relation to future myopia development. Most of these should be familiar by now, except for the accommodation items, which I won't explain here. This is the full results table from the article with the odds ratio for each variable. I don't want to push into a lot of detail here because it is the conclusions that are important. Note, visual activity in diopter hours is a measure of near work. The results for age 6 showed most of the factors had some relation to later myopia, but of variable strength. Near work had an odds ratio of 1. To see if they could improve predictive value, they also did multivariable analysis. Of the 8 significant baseline parameters, only baseline refraction and astigmatism were significant in all years. The author's conclusion of this large and well-done study was, the best predictor of myopia development was baseline refractive error. That single parameter had nearly the equivalent predictive strength of the other 8 variables put together. As a practical guide, they identified children who started out with a low amount of hyperopia as the most risk of becoming myopic. They specified cut points, signaling risk of future myopia. The concept is easier to see looking at this graph of myopia development. The family of curves shows that the earlier myopia starts, the higher degree of myopia you end up with. In considering treatment, if the aim is to prevent myopia over 3 diopters, according to this study, you would treat children whose myopia developed before age 8. Here is another version of the same graph strategy with somewhat different curves. In this study, if myopia started at age 8, by age 17 the average refraction was over -6 diopters. Looking another way, the chance of developing high myopia, defined as greater than -6 diopters. For those whose myopia started at age 7 or 8, in this study population, the risk was greater than 50%. So age of onset of myopia will be an important, perhaps key, deciding factor that we will talk about in the treatment video. It is noteworthy that the divergence from emmetropia toward myopia actually begins several years before passing the myopia threshold. This is covered in detail in video 4 on myopia divergence. We started this section by asking, what are the causes of myopia? We looked at studies that tried to identify genetic versus environmental contributions. To summarize our results, we can assemble a scorecard. There is general agreement about the association with number of myopic parents and outdoor time. However, the role of near work does not come to such a neat conclusion. Recall the expert point-counterpoint argument. We can expand reading and near work section by looking at other avenues of evidence. We have been discussing a lot of observational evidence, but theories of cause can be tested by other means, like animal experiments and treatment trials. These are the factors found to be associated with myopia. Each has a proposed mechanism connecting it to myopia, which are covered in the other videos. Since we are talking about the non-consensus about reading and near work, let's use that as an example. The most obvious mechanism would be accommodative effort. Way back in 1985, these researchers reported a set of experiments that tested the connection between accommodation and myopia. Working with monkeys, they lesioned multiple sites to interrupt the incoming visual pathway and the outgoing accommodative pathway. No lesion outside the eyeball affected their ability to induce myopia. This is noteworthy because it means eye growth control is contained within the eye, and because there is strong evidence against accommodation as a cause of myopia. Another way of testing connections is through interventions targeted at causes of myopia. The range of methods includes various glasses and contact lens options, therapeutic drops, and increased light exposure. These trials are covered in detail in video 5. Continuing with reading and near work, we will show a couple relevant examples. For reading/near work, again addressing the mechanism of increased accommodative effort. If that is the case, then reducing the effort of accommodation with some form of reading correction should reduce myopia development. The COMET study is one of several studies to put this theory to the test, here using progressive bifocals with single vision glasses as controls. At the end of 1 year, the progressive lenses reduced myopia a small amount, 1/4 of a diopter, with no additional reduction in the following 2 years. The authors pointed out that while this is probably a true effect, it was not enough to be clinically useful. Some years later, another trial was done using executive bifocals, also with single vision lenses as controls. This trial showed more of a response to the bifocal. Why should there be response to one kind of bifocal and not another is an unanswered question. Conflict of study results is a common feature of myopia treatments. In video 5, we take a longer look at the range of study results. Now is a good time to recap the genes versus environment question. Animal studies that relate to myopia are covered in other videos, but there is one interesting observation that fits here. This is refraction in humans over their lifetime. This is refraction in monkeys over their lifetime. Physiologically, their eye development is very similar to ours, just on a different time scale. Taking 1 human year as equal to 3 monkey years allows us to make some comparisons. If we take the human period we are interested in as up to age 30, there is a high proportion of myopia. The equivalent monkey youth would be up to 10 years. In that period, they show a few hyperopes and no myopes. Comparing the equivalent age groups side by side, for the monkeys, we do not know their history of light exposure. Presumably, they have little schoolwork and are unlikely to have glasses. Thus, to the extent we can connect monkey eye with human eye development, this could be taken as the direction of human eye development without the influence of myopogenic environmental factors. A compelling thought. For a broad view, we return to the 2018 Morgan Review paper of myopia causes. Note the panel of all-star authors. Here is how they analyze the world increase in myopia. There is an epidemic of myopia in Asia with attendant high myopia. High myopia having potential sight-threatening consequences. Because this increase has occurred over a relatively short period of time, it must be acquired, not genetic. They contend the contributing factors appear to be heavy emphasis on near focus and limited time spent outdoors. These risk factors should be modifiable. There are additional measures, for example atropine, that can be used to control myopia. A year later, Morgan and Rose provided a critical review of the genes versus environment debate, including historical background, study methods and conclusions, and current study results. These excerpts, I think, cover their conclusions. First, quote, environmental factors can have a major impact on development of myopia. In parallel, genetic contributions to variations in refractive error are more limited than has been generally assumed from twin studies. Second, quote, the idea that myopia primarily occurs due to a genetic failure of control over refractive development needs replacement. In sum, quote, school myopia should be regarded as the result of a control mechanism operating in conditions that are so unnatural that myopia occurs as a disease of advanced civilization. On that thought, we conclude the main section of video number 1. But before we arrive at our summary, there is a practical item of note. In the refraction section, we did not discuss presbyopia or astigmatism because they aren't important for myopia development. It is unavoidable that over everyone's lifetime, the lens inside your eye gradually loses its flexibility. At some point, usually in the early 40s, it becomes noticeably more difficult to focus up close. Thus, the need for reading glasses. For those who have myopia, that is functionally equal to having built-in reading glasses, a not insignificant benefit for myopes. Now, we are ready to summarize the key points of Video 1. Myopia is increasing around the world, particularly in Asia. We know that genetic background accounts for basic susceptibility, but it doesn't explain the whole picture. Researchers believe the prevalence is increasing faster than can be accounted for by changes in gene frequency. There must be other contributing factors. In our modern world, children spend more time indoors with less exposure to sunlight and less exercise. There is increased near work, as in studying and computer work. And more recently, there is a question whether there is a contribution from the way standard glasses correction works. Understanding the causes of myopia and how to treat it remains a work in progress. As we end video number 1 in our myopia series, if myopia has caught your interest, the next 4 videos cover more myopia subject in detail. Video 2 is a discussion of how myopia leads to vision-threatening complications like retinal detachment and myopic macular degeneration. Video 3 takes us into experimental investigations of myopia, trying to understand the remarkable ability of the eye to control its own growth. Despite having a mechanism for growth control, Video 4 explores how one group still diverges into myopia. We end with video number 5, exploring the variety of treatment options available and how successful they are in trials and their use in practice.